In this first practical, we're going to introduce you to some of the most commonly used species in photosynthesis research and see how they can be used as research tools to ask interesting biological questions. When studying biological systems in the lab, we often use model organisms. These can be defined as a species that is extensively studied to understand particular biological phenomena, with the expectation that discoveries made in the model organism will provide insight into the workings of other organisms. In animal research, commonly used models are the fruit fly Drosophila melanogaster and the mouse Mus musculus. In plant biology, the most commonly used model organism is a flowering plant called Arabidopsis thaliana, and you'll be using this species in the practicals. Other model organisms for studying photosynthetic life include the green algae Chlamydomonas reinhardtii, the liverwort Mercantia polymorpha, which is used as a model for the early land plants, and the crop plant Zea mays. Arabidopsis thaliana is a good choice of model organism for a number of reasons. It's a relatively fast-growing species. The entire life cycle can be completed in as little as 8 weeks under the right conditions. It's relatively small and it produces a large number of seeds, and it's easy to grow in the lab. The genome was sequenced in the year 2000, and that genome itself is 125 million base pairs long, which is actually quite small for a plant. It's quite easy to make mutant plants, i.e. it's relatively easy to disrupt the DNA in the genome and then determine the effect that this has on the plant. And there's now a large database of information about the species which is hosted online. However, Arabidopsis also has its limitations as a model. For example, it doesn't make any woody structures, so it isn't a very good model for trees. Unlike most plants, it doesn't make symbiotic associations with soil microbes, so you can't use it to study those processes. The process you are interested in scientifically determines your choice of model organism, so it's important to know the limitations of your species as well as its advantages. Using these model organisms, we can discover the relationship between the genotype of a plant and its phenotype. So the specific DNA sequence of an organism is known as its genotype, and then the phenotype are the observable characteristics. So in these practicals, you're going to determine the effects of a change in the genotype on the phenotype of your plants. So we're going to be measuring things like growth rate, chlorophyll content, epidermal development, stress responses to characterize the phenotype of your mutants. And then in the later practicals, we're also going to do a DNA extraction and genetic techniques to investigate the genotype of the plants. We're going to link the two things together. So in this practical, we're going to provide you with some Arabidopsis thaliana seeds that we ordered from the Nottingham Arabidopsis Stock Centre, and you'll be using plants that you set up in this practical in later practicals. Each group will be provided with 2 different tubes of seed. You're going to get some wild type seeds. So wild types are seeds that were originally collected from the natural environment and are not mutated in any way. And these act as a control for our experiments. There are various different wild types out there, but none of them are genetically mutated deliberately. We're also going to give you some mutant seeds. So these are seeds which have had their genomes deliberately manipulated, i.e. the DNA sequence has been altered in some way. There are 2 main ways in which we make mutant plants. So one of them is to use either X-rays or chemicals that causes individual bases in the genome to change, i.e. at a particular point in the genome, a G residue might be changed to a T residue. So this could occur at any point in the genome. If it happens in an important region of a gene, it could disrupt the function of that gene. And those are what are known as point mutations because it's an individual base that's been changed. Another way to mutate plants is to insert a relatively large piece of additional DNA into the genome, and that results in the production of a transgenic plant. It's got genes from different places inserted. So you could either insert some DNA that contains something useful, so you could insert a disease resistance gene from another species into your plant and then see whether its response to infection is different. And this would be known as a gain of function mutation. There's a new useful gene that have gone in. Alternatively, what you can do is to insert a non-coding sequence, which is known as a tDNA, and this can be inserted at a random point in the genome to make what's called a tDNA insertion line. So if that tDNA inserts in the middle of a gene, then that gene might no longer function, and that might have an impact on the way that the plant functions. The mutants you'll be using in these practicals are tDNA insertion, but you won't know which gene the tDNA has inserted into until we do some analysis of the DNA later on. We're going to be growing your Arabidopsis plants under sterile conditions, which means that you're going to have to use aseptic technique to avoid your experiments being contaminated with any bacteria or fungi in the environment. To do this, you're going to wash the seeds in ethanol and then you're going to expose them to a 50% sodium hypochlorite solution, that's bleach, to kill off any microbes on the outside of the seed. You'll then wash the seeds in sterile water to remove the bleach and then finally suspend them in a dilute agar solution to make them easier to handle. You'll then plate the seeds out onto agar plates using a commonly used plant growth medium. There are several tricks that you'll need to use to minimise the amount of contamination when you're working. So you'll do your work in the presence of a lit Bunsen burner so that any bacteria or fungal spores in the environment will be killed off. You'll be provided with pre-sterilised solutions and equipment. It's been through a piece of equipment called the autoclave that will sterilise it. And it will also be important to work quickly and to minimise the amount of time that your seeds and your agar plates are exposed to the air. So we'll give you advice on how to do this and how to work safely. Remember that you'll be working with genetically modified plants in these practicals, so you're also going to have to observe appropriate biosafety measures to stop the release of any GM material into the environment. So in the practical, there'll be various things that you need to do to sterilise your seeds. There'll also be demonstrations about different model plants and algae for you to look at and some question sheets for you to fill in. We're also going to give you some training onto how to set up and record your data in a lab book, so that'll be your personal lab book, and you'll need to submit this lab book for assessment at the end of the module. So keeping a good quality lab book is a really important skill. Before coming to the lab, you should make sure that you've read the lab manual that's available on Canvas, including the risk assessments, and submitted the pre-lab quiz for Practical 1 to check that you've understood what you'll be doing in the lab. So I look forward to seeing you in the lab to have a look at some of these model plants.